guess it is 11:15. So I'm Mysia. I am a postdoc in the Moran Lab at Texas A&M, and I'll be talking about some of my postdoc work on sex determining regions in darters. So, like I assume many of you in the room, I'm really motivated to study the relationship between speciation, genomics, and behavior. And in particular, I'm interested in the pattern of reproductive character displacement, where we have two species that are more different where they co-occur in sympatry compared to where they occur in allopatry. And a big question in our field is, why is this pattern here? And one potential explanation is that we have selection against hybrids that results in this evolution of enhanced prezygotic isolation, and this process is what we call reinforcement. And today I'm going to be focusing on this part of the process where we have that selection against hybrids. And one big motivating question is, what are the genomics underlying these selection against hybrids? And some potential explanations are things like chromosome rearrangements, such as inversions, where we got a part of a chromosome that does a little flip-flop and causes problems with chromosome pairing and crossing over during meiosis. We also have epistatic interactions like Dobzhansky-Mueller incompatibilities, where we have an ancestral genotype give rise to two daughter lineages, where they have independent new mutations arise, and these mutations become fixed, and then when they're brought together in the hybrid generation, they end up interacting and causing problems in fitness. So I'll be focusing on one group that's really fun to study these processes in. So these are the North American darters. They're an incredibly diverse set of stream fishes that are arguably very cute. I would say the cutest fish. I'll be focusing on two of these darters, the orange throat darter and the rainbow darter. And throughout the talk, the rainbow darter will be highlighted in blue, and the orange throat darter will be highlighted in orange. These two fish share color patterns, ecology, mating behavior, and they commonly hybridize. You can go out and catch a lot of F1 hybrids. So what's really interesting about this group is that we have one species of rainbow darter, Ethiostomus ceruleum, but we actually have an entire clade of orange throat darter species, which is thought to have arisen due to their occurrence in sympatry with the rainbow darter. And most of these species either occur in sympatry or just in allopatry. One exception to that is Ethiostomus spectatile, and that's going to be the orange throat darter I focus on. It's the only orange throat darter that occurs in both sympatry and allopatry. And what my postdoc advisor has shown is that where the orange throat darter and rainbow darter co-occur, we have reproductive character displacement because sympatric males strongly prefer females of the same species, whereas allopatric males do not show a preference. But is this also an example of reinforcement? So looking at hybrid fitness, we see that when hybrids are back-crossed to the parental species, they experience lower proportion of eggs fertilized and hatching, and hybrid crosses are disproportionately male despite having the same clutch sizes. And so therefore, we have good evidence that this is a great example of reinforcement. So we have range overlap, gene flow, and postzygotic isolation. And we can ask, what is driving this postzygotic isolation? And we have a lot of reason to think, we have a lot of potential explanations, including chromosome structure, which varies a lot between and within species. So this is the orange throat darter and rainbow darter karyotypes. You can see a lot of variation. And looking within a single species and different populations, you also see a lot of variation. We also know that there's widespread divergence and rearrangements between the orange throat and rainbow darters. So this is a linkage map, and you can see a lot of places where there's potentially inversions and movement through the genome. And if you look at DXY and FST across the genome, it's kind of spread out across the genome. But today I'll be talking about this one reduction of hybrid fitness where we have that disproportionate male in hybrid crosses. And so we want to know if this is evidence for an incompatibility in sex determination. And part of why we want to study this is that sex-determinant fish is incredibly diverse. We have XY and ZW systems, we have polygenic and monogenic systems, and a lot of different potential genes. So we want to know if these two species have different sex-determining systems. So the questions I'll be addressing today are, where are the candidate sex-determining regions in these species? What are some candidate genes? What evidence is there for differences in sex determination? And are these candidate sex determining genes or regions in places where we've already identified genomic incompatibilities? So to address this, we sampled 12 male and 12 female from both allopatric and sympatric populations of both species. We did whole genome resequencing. We processed that data with the standard pipeline, and then we conducted 3 separate analyses. So our first approach is a population genetics approach. So we calculated intersex FST by site and by gene for just sympatric populations. And we identified the top 5% FST by gene outliers and performed a gene ontology analysis and identified some candidate genes for sex determination. And then we looked at the FST by site to see if there's any part of the genome that has particularly high differentiation between the sexes. We then did association tests, so we just did linear mixed models in GEMMA, and this was for pooled populations for sample size reasons. We then took a depth of coverage approach in the sex determining regions where females have, for example, XX, whereas males have XY. We expect differences in coverage in that area between males and females. So we calculated the depth of coverage in 10 kilobase windows using bed tools. And then we normalized the coverage and estimated the difference between males and females. So what did we find? So kind of looking at where we have high differentiation genes in these genomes, we had 604 FST outlier genes for the orange throats and 996 for the rainbow darters. And only 10 of these were shared between the species, and of those 10 genes, only one of them is potentially involved in sex determination hormones, and that's ZBTB7A. More on that in a little bit. Looking for what ontologies were enriched for each species within that set of candidate genes. For the orange throats, we had male sex differentiation, anti-Müllerian hormone signaling, negative regulation of androgens, response to luteinizing hormone, regulation of gonad development, and spermatid differentiation. Whereas in the rainbow darter, we had pituitary hormone function, regulation of sperm motility, and sperm axiom assembly. So we have some evidence here that there's some distinct gene functions involved in sex determination across the 2 species. Looking at the actual list of genes and comparing that to what we know about sex determination in fish, we found that within the set of FST outlier genes for the orange throat, the anti-Müllerian hormone on chromosome 9, TGF-BR3, which is in that same family of proteins, and that ZBTB7A that I mentioned before. And all of these are found on chromosome 9 in orange throat, and that's going to come back in a minute. Whereas in rainbow darters, we had another gene in that same kind of family as AMH and TGF-BR3 that's been identified for sex determination in other fish species, found on chromosome 18. Yet another gene in that whole family on chromosome 18, and then that shared one with orange throat on chromosome 9. Getting into the FST by site, we looked at where we have high levels of intersex differentiation across the genome. And in the orange throats, we see really high levels of intersex FST, so high levels of differentiation on chromosome 9, which is where those candidate genes are located. Whereas in the rainbow darters, intersex FST values are kind of interspersed across the genome, and there's no clear pattern the way there is in the orange throat darters. We also conducted those association tests on those pooled populations, and yet again, chromosome 9 is coming out as a strong candidate region in the orange throat darter, whereas in the rainbow darter, we now have a strong association between sex and sites on chromosome 23, which is not where the candidate genes that we previously identified, so that's something interesting. And then the final approach we took was that depth of coverage approach. And spoiler alert, we haven't found any clear patterns. So I'll show you some results from these chromosomes that were implicated by our other analyses. So these plots will show you the differences between male and female coverage. This gray area here is showing kind of the genome average. And so if there were differences in coverage, we'd see high levels of variation. So for chromosome 9, which again was implicated in the orange throat darter, we don't see any patterns of differences in coverage. And for the rainbow darter, same thing. You'll note that this kind of looks a little messier, and that's potentially because we have aligned it to the orange throat darter genome, which we're going to readdress later. Looking at chromosome 18, which was implicated by candidate genes in the rainbow darter, we don't see anything in the orange throat darter, and there might be a little trend in the chromosome 18, but absolutely not clear or a big enough difference that we are looking at it yet. We also, chromosome 23, which had this strong association with sex. Something is going on at the tail end of it in the orange throat as well as the rainbow darter, but we're not entirely sure what's going on there. These are still preliminary results. So that's definitely an area where we want to look into and kind of see what's going on. So to kind of address the questions that I laid out for at the beginning, we have some candidate sex-determining regions on chromosome 9 in the orange throat darter, whereas we have candidate regions on chromosomes 18 and 23 in the rainbow darter. Some candidate genes are the anti-Müllerian hormone and TGFBR3, both on chromosome 9 in the orange throat darter, and we're really interested in TBC1D32 on chromosome 18 for the rainbow darter. And so this is some preliminary evidence that there are different sex-determining regions and genes involved in sex determination for those sympatric orange throat and rainbow darters, which could be leading to our male-biased hybrid clutches. And then we also wanted to know if these correspond to areas that we've already identified as areas of genomic incompatibilities. And we do see that chromosomes 9 and 18, which are where our candidate genes are located, and where we had strong sex association and differentiation in chromosome 9 in orange throats, do have previously identified inversions. So that is that orange chromosome over there and the green one at the end. And there's, if you look closely, which you might not be able to see, there seems to be some inversions there. So to kind of tie this into what we know about other fish species, the anti-Müllerian hormone pathway is implicated in a lot of different species, including yellow perch, which is closely related to darters. One of the receptors has a duplication, and it's in close proximity to AMH on chromosome 9. And it also functions in other fish species, including grouper, tilapia, and stickleback. And then the European perch, again, same family as darters, has a large sex-determining region on chromosome 18, which includes a lot of genes in the same family as AMH, including TBC1D32, which is one of the genes that we found to be important in rainbow darters, so that's exciting. And kind of future analyses, we're going to do some SNP density analyses, we're going to revisit the depth of coverage, and then look at allopatric populations between species, and then allopatric versus sympatric within a species. With that, I'd like to thank my lab and our funding sources, and I'll take any questions. Yeah.
Did you map the variants that you came up with to see where putatively this gene they actually mapped to?
We haven't quite gotten to this yet. This is like right off my computer last week. But yeah, that's kind of the next step, is to kind of see where those genes are, and if those genes, we're going to zoom in on the FST and association test results within the chromosomes of interest and see if those align with where the genes are. We're also going to look at heterozygosity to see if there's any differences in heterozygosity between the sexes in those same regions.
Very cool.
Any other questions? Yeah, sorry, I didn't see you.
Do you know how closely related the two species are?
Yeah, they're pretty closely related. They are within the same kind of group. So yeah, they're pretty closely related, and I am trying to help the next person. Okay, there you go. Sorry. Okay, thank you.
Hi everyone, thank you so much for being here. Today I'm going to be talking about exploring genomic divergence between two sympatric stickleback ecotypes. These are results from the first chapter of my dissertation. I'm a second going on third year PhD student in Kieran Samek's lab at the University of California, Riverside. And before I get started, I'd like to thank a couple people on my author list. First, Allison Bell in her lab. Allison gave a really awesome talk on white stickleback parental care behavior the other day. That was really cool, and I highly recommend you checking out her talk. I'd also like to thank Freddie Chain and Michael Olafume. They did some really cool copy number variation analyses on these data, but unfortunately I won't be able to talk about those results today, but happy to answer them later. Okay, so obviously we're all interested in evolution. I don't have to talk too much about this, but yeah, a central goal of evolutionary biology is to understand how biodiversity originates and how species boundaries are maintained over time. Three-spine stickleback are a classic system for understanding speciation dynamics, and this is because three-spine stickleback are ancestrally marine, and since the last glacial maxima, they've repeatedly colonized freshwater environments, leading to the evolution of several of these different ecotypes, many of which co-occur. I'm going to be talking about one of the lesser-known species, stickleback species pairs from Nova Scotia, Canada. These are called the white stickleback. White stickleback are an especially young species pair compared to some of the other species pairs that are much more diverged. And young species are particularly useful for understanding the cause of speciation. All right, so white stickleback, like I said, are from Nova Scotia, Canada. They are anadromous, so they spend most of their lives in the ocean. No one really knows what they do. But they come back to these brackish lagoons in Nova Scotia during the breeding season. And so this is showing a white stickleback male on the top and a common stickleback male on the bottom panel. Whites are generally smaller, or the males are generally smaller, and they clearly exhibit this pearlescent nuptial coloration that you don't see in the common male, which is larger. So the white stickleback was originally discovered by Max Blau, and yeah, he noticed the white males were smaller. They have this striking nuptial coloration, but he also noticed that they only nest exclusively in filamentous algae, and they've completely lost their parental care behavior, which is so well known in stickleback. So they're kind of like the terrible fathers of the stickleback. And based on this suite of really interesting characteristics, Max Blau speculated that these might be a new species. So during his PhD, my advisor, Kieran Samick, revisited the white stickleback system. And so he gathered rad-seq data from across the range of white and common stickleback in Nova Scotia. And he basically found that white and common sticklebacks form genetically discrete populations. What was also really interesting is he found that genome-wide FST is extremely low, so it's around 0.02. He also did some pretty intensive morphological and stable isotope analyses, and he found that there are no significant morphological and isotopic differences between the ecotypes. So this leads to the pretty obvious question of what is keeping these white and common stickleback from collapsing into a single hybrid swarm. And so there's a host of pre- and post-zygotic barriers we can think about, like female mate choice, male nest site preference, so the whites only prefer filamentous algae. Obviously, the whites have also lost their parental care behavior, which is really interesting. And we do find that these populations will hybridize in the wild, but they don't seem to make it past the F2 generation. Something we also don't know about is what is the evolutionary history of white and common stickleback? So are these actually two populations that have come into secondary contact? And when do we think they diverged? So Kieran's previous work was limited by rad-seq, so we wanted to revisit the system using higher resolution whole genome sequence data. And so we acquired whole genome sequences from 10 males and 10 females of each ecotype, so 40 individuals total, from 2 different locations. And we got whites from one population and commons from another population, as you can see in the map. My lab mate, Gina Lucas, prepared the libraries, and then they were sequenced at 80x coverage. These were then aligned to the most recent stickleback reference genome, and yeah, that's it. All right, and very conveniently, we found that with whole genome sequence data, whites and commons also form two distinct genetic clusters. And also conveniently, genome-wide FST is also 0.02, just like with the RADSeq data. To further rule out that the patterns that we're seeing were not due to geographic distance and are actually due to reproductive isolation, we ended up acquiring an additional 17 individuals from Paul Benson's lab at Dalhousie University, and these included 3 other locations where white and commons occur in symmetry. These were then sequenced at 30x coverage, and we also acquired another population from the St. Lawrence River in Quebec. And really conveniently, additional whole genome sequences from additional populations continue to support the existence of these distinct genetic clusters. So we can see Nova Scotia whites, commons, and the Quebec common are each their own distinct little group. So to further explore the genomic landscape of divergence between whites and commons, I performed a population branch statistic test. So PBS is very similar to FST, but instead of looking at pairwise genetic divergence, you can include basically like a third outgroup population to look at lineage-specific differences. And by computing PBS branch lengths, one can look for regions that might be under selection. And so this is just showing the PBS scan of the Nova Scotia whites. And unfortunately, we didn't see, there's not one smoking gun or a clear region of differentiation. Instead, we see near fixed differences kind of across the genome. And so there's clearly many outliers, and what you might be thinking, what are some of these outlier genes? One of them is SOX5, and this is a transcription factor which is responsible for determining whether pigment cells develop into leucophores, which are white chromatophores, or xanthophores, which are yellow chromatophores. And the picture below is just showing a study that was done on Medaka, and the loss of SOX5 causes an excessive formation of leucophores and a total absence of xanthophores. Another outlier gene was ESR2A, which is an estrogen receptor. ESR2A is essential for normal egg development and obviously is really important for female reproduction. And this is really important and interesting for this system because if you saw Allison's talk, there are known differences in egg morphology and also ovarian fluid between white and commons. And so the picture is just showing a study that was also done in Medaka, and on the left, right. Yeah, so those eggs develop normally, but the ESR2A knockout individuals do not form proper eggs, and those do not get properly fertilized. All right, so the other thing that we wanted to address with this project is what is the evolutionary history of white and common stickleback? And to get at this, I performed demographic inference using the command line interface version of DADDY, and I attempted to fit 11 different demographic models. I found that the best fitting model was a model of divergence around 30,000 years ago, followed by 2 periods of high gene flow. So basically this symmetric migration model in 2 epochs. And this date actually it's not that old if you study other systems, but it's kind of old to me for stickleback. But it actually coincides with the timing of diversification of the major transatlantic stickleback clade. So perhaps there's been multiple invasions of stickleback populations to the Western Atlantic from Europe. All right, so in summary, white and common stickleback occur in symmetry during the breeding season, but they exhibit really low levels of genetic differentiation, so FST is 0.02, despite them being very phenotypically and behaviorally distinct. Our genome scans also identified several candidate genes that might contribute to reproductive isolation, but these have not been functionally validated just yet. And finally, my more preliminary demographic modeling suggests that white and common stickleback have experienced high rates of gene flow since they initially diverged around 30,000 years ago. And I plan to further fine-tune these demographic models and also look at population size history using programs like PSMC later on. And with that, thank you so much for your time, and I'm happy to take any questions. Thanks to everyone who helped. Any questions? Yes.
Yes, really interesting talk. I'm wondering, has anyone looked at the behavior differences, if there are any, in the anatomy of the white and common stickleback? Do they arrive in the freshwater system at the same time, and do the young leave at the same time?
Nobody knows, but if someone wants to study that, I'd love to help them because I'm also very curious.
Thank you.
Yes.
Was the ADX coverage, is that for individual or is that for the whole group that you see?
I believe it was for each individual, but I could also be wrong.
Are you happy with that coverage?
Yes, it's really nice.